My name's Michelle, I'm a parasitologist, and our research focuses on looking at the interactions between parasites that are found in humans, wildlife and the environment. And this is the field, I guess, of trying to understand disease ecology. We work on a whole range of different parasites, including those that are found in the gut, such as Cryptosporidium and Giardia, and also those that are found in the blood, such as trypanosomes and the parasites that cause malaria. And we work on a whole range of different hosts that sees us getting out into the field. This includes hosts that are in the marine environment, such as the little penguin and Australian sea lions. And on the land, we have major projects looking at possums, echidnas, Tasmanian devils, flying foxes, a whole range basically of different wildlife species. And this is mainly because we're interested in the ecology of the organisms that we're looking at and how they're being transferred between each of those wildlife hosts and people. Over the years, we've worked on a number of different systems, hosts and parasites, and thought about how they interact within each other. So traditionally, one would look at one parasite and one host. Our research has gone beyond that, and we're looking at interactions. So we're trying to understand co-infection with multi-parasite species, and also how the endemic microbiota, so the bacteria that you would find in the gut, as an example, interact with those invading parasites. More recently, we've been looking at the transfer, or what we might term reverse zoonosis, the transfer of human-associated organisms into wildlife species. And we've been particularly focusing on antimicrobial resistant bacteria. These bacteria have overcome, or have gained the ability and overcome the use of antibiotics or the medicines that we use to treat them. But the whole ecology of these organisms has now changed, and they've been disseminated from humans into the environment and into our wildlife species. And we're trying to understand the impact of those antimicrobial resistant bacteria to our wildlife. In addressing this question, we have a large citizen science project in collaboration with Taronga Zoo and also Inspiring Australia. And we have all sorts of citizens trekking into their backyards and collecting us a possum scat sample using a special kit. That sample comes back into the lab where we apply a range of techniques, including traditional culture techniques, so we will culture the bacteria, and then also apply genetic techniques and DNA sequencing to determine if the bacteria are resistant to antibiotics and what they're resistant to. This research is particularly significant because it's giving us an understanding of the depth of antibiotic resistance in the environment and contributing towards understanding antimicrobial resistance ecology and antimicrobial stewardship, and hopefully towards saving these important medicines for treatment of humans and also wildlife when those treatments are required. Working with wildlife requires working with a lot of different partners, and our partners are stakeholders in the research outcomes that we're producing. They include WIRES, Taronga Conservation Centre, and also the citizens that are contributing those samples through our Scuba Poop project. The outcomes of our research investigating host-parasite interactions between wildlife and humans is critical for understanding infectious disease ecology. So if you're interested in blending fieldwork with laboratory research and addressing critical questions that are relevant to future health, get in touch.